So this is a talk I gave at the 2024 Gordon Conference at Mount Holyoke College. And there are a couple of things that you should know. First, Chris Miller appeared the day before my talk and was around and was his usual self, so I make a mention of Chris. And actually, the talk before mine was by Stephen Long, who also figures prominently in what I'm going to talk about. I was in between people and beer time on the last night of the meeting, so it was kind of a fraught occasion. That evening, Shiva Mandala had presented some really nice cryo-EM work on a voltage-gated channel under a membrane potential, so that when it came to give this talk, I said, well, you know, Shiva has given much better data than I have, so I have to make sure that, there we go. I'm going to talk about this. I'm going to talk about a kind of personal view of how the whole idea of a voltage sensor and what its structure is and how it worked has come clear during the time that I've been able to watch it. It's been my privilege to watch this progress in science. So we'll start way back when I was a college student. I was a college student at Caltech, and there were two people who sort of helped me, an applied physics major, learn a little bit about biology. Henry Lester, who of course was very interested in neurotransmitter receptors, and I took a biophysics class from Max Delbruck, and that was a real eye opener for me. One of the things that we did in his class was we read one of Chuck Stevens' papers where he showed power spectra computed from the fluctuations in current through an acetylcholine receptor channel. And I said, oh, I know what a power spectrum is. Hey, this is way cool. So I wound up going to graduate school in Chuck Stevens' lab, and that was a wonderful experience. He said, well, yeah, there are just two problems in the field of ion channels, and you can see ion selectivity and gating, and I thought gating was way cool. So Chuck said, well, you've got to go read the Hodgkin-Huxley papers. So I read them, and right away I saw, wow, this equation, well, that's just the Fermi-Dirac distribution of electron energy levels. Wow, I recognize that. And of course, this is the point in Hodgkin-Huxley's last paper where they're pointing out that the voltage sensitivity of voltage-gated channels is really steep. See, because I was used to transistors, which had this particular voltage dependence that's set by the charge on an electron, and these channels had this incredible steepness. And so we know, for example, from the Fermi-Dirac distribution or Boltzmann distribution, that means that whatever is sensing the membrane potential has to have a lot of charge. Uh, and that raises an interesting question, which is how do you make a protein that senses membrane potential and has so much charge movement? Well, in those days, not everyone was convinced that the channels existed at all. So a few years before was the first paper that I know of that presented the idea that sodium current is carried by just a few sites in the membrane. This is an amazing experiment. So they had a chamber. They dissected out, I think, 7 walking leg nerve bundles from a lobster. And they had this little 50 microliter solution here with 300 nanomolar tetrodotoxin. And what they would do is they would put in one nerve bundle, measure the compound action potential, put in the next, put in the next. And the idea is that the tetrodotoxin would bind with high affinity to whatever was carrying the sodium current, and it would get depleted as they went from one nerve to the next. So here's the very first nerve bundle, and you can see that as soon as they put it in the solution, the action potential decayed very rapidly. But then by the time they got around to the 7th, they were not blocking as much of the action potential. And from that, they were able to put a lower bound, I'm sorry, an upper bound of 13 sodium channels per square micron density on the lobster axon membranes, which was astonishingly small. It said that somehow the sodium current is not a general membrane phenomenon, but it was just carried by a few sites. Amazing. So my own PhD thesis was on counting sodium channels. And so I used this method where you would give one depolarization after another, and you would measure the sodium current, and there would be, if you subtracted off the fluctuations, you could see these little variations, which had to do with a random number of sodium channels out of the population opening each time, and that gave some that gave fluctuations that could be analyzed, and we could count how many sodium channels were there or estimate that. Now, while I was in Chuck's lab doing this, Erwin Neher was visiting, and he and Bert Zachman had worked out this really pretty heroic experiment where they would voltage clamp a frog muscle fiber and put this, what we now call a loose patch clamp pipette, up against the cleaned membrane of the muscle fiber. And this was Erwin's first recording of single channels of acetylcholine receptors. I then was very fortunate. I was really fortunate to go and do a postdoc in Erwin Neher's lab. And this is a typical picture of Erwin. He's at the setup looking into the microscope. Erwin was the sort of person who, he was director of a department, so he had a corner office on the third floor of our building. But, he went and moved into a much smaller office and made his big corner office his laboratory and moved his setup in there. This is the amazing experimentalist who is always thinking. So the group that we had in Erwin's lab was Alain Marty and Erwin and Melanie Knudsen and myself, and Alain, of course, is showing off the magical pipette that makes the GigaSeal possible. So you probably have seen this this paper, the GigaSeal, the GigomeSeal paper for doing high resolution patch clamping. You'll notice that my name is at the end of the author list, which actually does not mean anything because in those days, it was good form to put the names in alphabetical order. And so I was the lucky one with the last, with the lowest in the alphabetical order. And there was lots of fun in Göttingen there in the early 1980s. I think one of the most exciting days was the day that Alain Marty first saw repeatedly the huge BK potassium channel current. This was a big deal because in those days, people said sodium channels have a small conductance, like 10 picosiemens, and that's because they're a selective channel. And nicotinic acetylcholine receptors have a large conductance, 25 picosiemens, and they can have a high conductance because it's non-selective. So then Allah sees for the first time this highly selective potassium channel with a conductance of like 200 picosiemens. And so here is everyone gawking at this. Bert Zachman with his mouth open, David Cahoon smoking his pipe, Aaron and Allah. A wonderful occasion. Now, also around this time, Shisako Numa and his cast of thousands did the amazing job of cloning the cDNA of the sodium channel, which is, as we know, one of the longest genes for ion channel that we know of. And already it became clear that there is this segment that they called S4. It's a sort of the fourth alpha helix that was visible, and it had this repeating every third residue being an arginine or a lysine in each of the four homologous domains. And in their first paper, they said, well, this must be an alpha helix that's not in the membrane. It's the one that has all these positive charges. But soon thereafter, people said, oh, this must be a voltage sensor. And various mutations confirmed this. Then a few years later, there was a remarkable year. Newton had his miraculous year in 1966 when he, 1666 when he worked out gravity and calculus. Einstein had his miraculous year in 1905 when he did the photoelectric effect, Brownian motion, and special relativity all in 1 year. Well, okay, so in 1990 was the miraculous year for potassium channels. And of course, Chris Miller is the one who, well, he published the paper, but the work was done by various people. At that time, well, the Shaker potassium channel had been cloned for, I think, 3 years before. And people had worked out, well, there is just this very short sequence here that somehow made the selectivity filter of the Shaker potassium channel. And Bill Zagata, Toshi Hoshi, and Rick Aldrich had worked out how inactivation worked with this ball and chain model and the ball peptide being able to reconstitute inactivation by just adding it back on the intracellular side of a membrane patch. Now, I mentioned before this whole problem of where does this huge amount of charge come from? How big is the charge in the first place? And one way to estimate the amount of charge is to do a so-called limiting slope estimation. That is, what you do is you go to a very negative potential such that this term vanishes and it just looks like this exponential function. And then if you plot the log of the open probability as a function of potential, you'll get the value of Q, the total amount of charge. The other way to see what this charge is to measure gating currents. And so in the same way here, if you have an electrode here and an electrode here, you can see the current flowing through the channel when the channel's open. But if you block the channel, you still see a current flow. When you change the membrane potential and force this charge to move, you will see a so-called displacement current that is the gating current. So we were fortunate, that is Nathan Chopa and Ken McCormick, were fortunate to be able to express a high level of Shaker potassium channels in a cell so that then we could measure macroscopic currents in the patch. And so the idea was we used the same kind of fluctuation analysis that I'd done for my PhD, in this case, to estimate the number of channels in a patch. And then we block those channels and measure a gating current. And so we could actually get an absolute calibrated value of charge movement based on the gating current measurement. And we came up with about 12 elementary charges per channel. And other people did this better and more completely. Uh, Sanjay Agarwal and Rod McKinnon did a heroic biochemical approach where they actually were able to count in a scintillation counter how many toxin molecules were bound to an oocyte and compare that with the total amount of nonlinear current which flowed in the oocyte membrane. And so they were able to then count how many billion channels they had and how many billion elementary charges were flowing, and they came up with a better estimate that was about 13.6 elementary charges per channel. And at the same time, they did this not only with the wild type channel, but also with mutations, testing, well, these various arginines and lysines, are they actually part of the voltage sensor? And the answer was yes, because if you mutate them, the total charge goes down. Now, Pancho Bezania, who actually was the one who first recorded a gating current, in his lab, they used an approach like ours, where they used fluctuation analysis to measure the total amount of current, and they were mutated various residues in the voltage sensor domain, and yes, those residues in S4 are the important ones. Now, the most amazing estimate of the charge movement was actually done in Joe Patlak's lab. So they were looking at sodium channels that they had mutated to make them non-inactivating, and they were able to get macroscopic currents of hundreds of channels in a patch. And their idea was, well, let's measure the maximum current we get from this large population of channels to estimate the number of channels. And then let's go to really, really negative potentials. Oops, I'm sorry. We'll go to really, really negative potentials where the open probability is small. And so what we're seeing here are occasional openings from a population of hundreds of sodium channels. And so from this very clever experiment, they were able to measure the open probability here over a range of 7 orders of magnitude. Now, the reason for doing this is to do this limiting slope experiment. You want to go to negative potentials where the open probability is very small so that that exponential term becomes really small, and you don't know when you've gotten to that really negative potential unless you really try it. So they did it, and they saw that they had this very nice constant slope. And this slope was also about 12 elementary charges. Now, this is very interesting because this is an entirely different method than measuring the gating current. And this says not only is the charge movement really big, but it's also very tightly coupled to channel opening. Channels won't open unless all of those 12 or so charges have moved. So that was really interesting and I think extremely clever experiment. Well, in my lab and also before us, Bill Zagata and Toshi Hoshi worked out kinetic schemes based on various kinds of measurements for how we thought that the voltage gating and Shaker potassium channels work. So this is our scheme, which says we have 4 independent sort of voltage sensing units, and then a couple of concerted steps before you get to the to the open state. And you know, when we were doing kinetic schemes, we thought that we were somehow learning something about the structure or the mechanism, saying, well, there are independent charge movements, and then there's something concerted at the end that actually causes the channel to open. And you know, it's now looking at it, we got some things right, which is that there were individual steps that had on the order of one elementary charge moving at each point. Now, the other thing in working out channel kinetics is, well, it's hard. It's hard to work out kinetics from single channel recordings. And a wonderful double E student, Lalitha Venkataramanan, spent some time in the lab working on so-called hidden Markov models. This is a mathematical technique that was first applied to channels by Peter Gage in Australia some years before. And the idea is we suppose when we make a patch measurement of single channel activity, underlying the activity is some kind of opening and closing of a channel. So in this simulation, this is just a two-state scheme. But what we see is that corrupted by noise or even more noise. And the interesting thing about the hidden Markov model theory is it says, suppose we want to know the rate constants. We don't really care exactly when the channel opens or shuts. We only want to know what the overall kinetic scheme is. And so this theory says, well, you know what? We don't care what the exact realization of the time course was. We'll just sum over all possible time courses and compute the probability of seeing what we see according to the values of the rate constant. So in this case, where there's not very much noise, we can converge pretty nicely using the so-called EM algorithm on the correct values of the rate constants. But here, when the noise is hopelessly large, we actually get pretty close to the right value of the rate constants through this trick. So this is the theory that's used in the well-known Cube software, for example, developed by Fong Chin in Buffalo. So then I took a sabbatical in Rod McKinnon's lab. That was a chance for me to do some remedial learning of biochemistry, and I think my main accomplishment in his lab was to slow down the KCSA structure determination. But the other thing I did while I was on that sabbatical is I commuted back and forth between New Haven and New York, and on the train I wrote a paper. And this paper had to do with applying the same idea, this idea of computing the likelihood, in this case, for the image alignment problem in cryo-EM. And here it's a similar kind of problem. What we want to do is we want, from some extremely noisy images like this, we want to know what the underlying true image or true structure is. And we don't care, even though an individual noisy image contains the actual motif rotated and translated, that's what this R operator is, by a random amount. We don't really care what those orientation and translations are. All we care is trying to determine what the true underlying image is. And so the wonderful thing about this likelihood calculation is that we just integrate over all possible orientations and translations to compute this quantity, the likelihood, and optimize it. And so that's what's happening here through hundreds of iterations of the maximum likelihood algorithm. And the really nice thing about this approach is that it avoids something that is famous in the cryo-EM field called the Einstein from noise problem. I'll tell you why it's Einstein in a minute. But the basic idea is, suppose we had a whole lot of pure noise images and we went and tried to align them to a reference that has nothing to do with atomic structures or anything. So if we keep aligning and averaging the aligned images, we come back with the same reference, the same motif that we started with. So if this were a picture of Einstein, then we would say here, well, we got Einstein from noise. Well, the maximum likelihood approach is really nice because instead of getting Einstein out, we just get noise out if we just have noise going in. So I'm really happy. I was really pleased that this bit of mathematics worked. So I mentioned before Pancho Bezania, who unfortunately is not at our meeting this year, who first, it was the one who first measured gating currents and has pursued the function of voltage sensors probably more energetically than anyone else in the field. And here's one of his many amazing experiments. This is one where uh, by replacing one of those arginines with a cysteine, he was able to bind a fluorophore in that position on the S4 helix and ask how much electrochromic shift due to the electric field do we get in the fluorescence of this fluorophore? And the answer is there's a really big electric field here. I mean, we're talking about 10 millivolts per angstrom as the magnitude of the electric field. And so that's giving us a bit more of a picture of what the underlying structure might be. Well, around this time, Steve Long gave us now a very nice high resolution crystal structure of a potassium channel, this KV 1.2. And if you've perfected the trick of looking at a stereo image wall-eyed, you can see in beautiful 3D where all of these residues are. But the point here is this is the voltage sensor helix bundle, and here are R0, R1, R2, R3, R4, the various arginines. Here is a very interesting, completely conserved phenylalanine, and then here's a lysine, another arginine, and here are various basic residues which keep those. Here are various acidic residues which keep the basic residues happy. So Steve Long and colleagues obtained this structure. This is the 4-helix bundle of the voltage sensor domains, and here are these arginines with their side chains looking up. And the idea, of course, is to imagine that the S4 helix slides down, and having those arginines, you can't see them very well, they're pointed now to the intracellular side, and this could be the motion that transports a large amount of charge across the membrane. Now, what about that phenylalanine? So again, in Rod McKinnon's lab, people look to understand what this phenylalanine is doing, and it's basically a gasket. It's the thing that keeps a tight seal so that not everything can just leak through this voltage sensor, but in effect, it makes a place where arginine residues get transported from one side of the membrane to the other. And so Xiao and Alice and Rod McKinnon would say this is a charge transfer center. An arginine that is passed by this phenylalanine is nicely coordinated by an aspartate and a glutamate down here, and this is a nice stable place for the charge to sit after it has been transferred. Okay, so we all have a nice idea of how the voltage sensor works in the sense that, well, we've seen it in the up position. That is, we have seen crystals and now also cryo-EM structures obtained with the channel in detergent or in a nanodisk, but anyway, in some kind of system where there is no membrane potential, and we always see those arginines all pointing up. So can we somehow get a structure with those arginines down, that is on the intracellular side? So this is the experiment that we were trying to do in my lab, and this is actually the same idea as what Shiva just told you, and Shiva, of course, has gone a lot farther and has much prettier data than we, but I want to show you ours anyway. The basic idea of the experiment is that we have our favorite channels reconstituted into small liposomes, and this is in a cryo-EM specimen. There's vitreous ice here, and there's a graphene support. And so what we're going to do is we will replace the buffer just before we freeze this. And hopefully in replacing the buffer, we make a potassium gradient. We have valinomycin around. We also have a potassium channel around. We have a potassium permeability, and we ought to make a membrane potential. I mean, a difficulty is that this experiment requires small liposomes, and the liposomes have to be small because they have to fit in a cryo-EM ice layer. And that means that to make our 120 millivolts, for example, all we have to do is transport 36 potassium ions out of this liposome. So we make these liposomes, we put them on a cryo-EM grid so that they are on graphene that is spanning one of these holes in the carbon film. And the proteoliposomes, of course, are made by purifying protein, solubilizing lipid, mixing them together, and removing detergent with, in our case, with dialysis. Okay, so here's a picture of one of our fields of liposomes, and in our particle picker, what we do is we subtract away the signal from the actual membranes of the liposomes, and then we find things that look kind of like a KV 1.2 potassium channel, complete with its beta subunits, and and that's what we work with. So the trick in doing the subtraction is fitting and subtracting a nice model of the membrane of a liposome. So here's an image of a liposome that's a model that we make, and we subtract off the model. Works pretty well. So then we accumulate a large number of these horribly noisy cryo-EM images, and then we just feed it to the black box in the lab. The black box in our case is this extreme gaming workstation. That would be the desire of any high-end computer gaming with 4 high-end graphics processors to do all of the number crunching. So we get a map. These are slices. These are projections through a 3D map, and this is the map that we get from the images where we've subtracted away the membranes, and then we put the membranes back, and you can now see here's the transmembrane region. Here is the curved membrane of a liposome, and these are the beta subunits in this wonderful intracellular domain that helps us find the particles. So we have a bunch of software. We mainly use the maximum likelihood program Relion and some special programs to deal with the liposome geometry. And we try various ways to try to make this membrane potential. And it turns out that never worked. It never worked, even though we tried sodium and potassium. We tried divalent ions. We tried impermeant anions and cations. Nothing seemed to work to give us a structure with a membrane potential. And the solution turned out to come to us from Rod McKinnon's lab, from Shiva's work, where they successfully saw a movement of the voltage sensor only in channels that had no potassium conductance at all. So they saw voltage sensors, they had channels where voltage sensors existed, but there was no ion transport. So of course, so we said, okay, all right, we better do that too. So it was already known that there is a mutation that makes a strongly inactivating channel, this W366F. This is the equivalent of the very famous Shaker W434F mutation. And people have now gotten cryo-EM structures of these channels, and the entire selectivity filter region is completely remodeled. And we used actually an enhanced version of this that inactivated very strongly, but also expressed well with 3 mutations. And so yes, so we could get inside out and right side out copies of our channel protein. So these are right side out. This is an inside out protein, and we actually got inside out and right side out at the same time. And you can think, well, the inside out one is going to be experiencing a positive potential if the right side out is experiencing a negative potential in this system. So the computers can, in their alignments of particle images, tell us what's inside out and what's right side out. And so here's a inside out structure. Here's the transmembrane region. We can tell that this is an open channel. For those of you who know are familiar with these things, that's the S4 helix. This is the S4 S5 linker. There's S5, and so we are seeing the S4 S5 linker pulling the channel open. So this is an inside out channel. This is a channel that is supposedly experiencing something like plus 100 millivolts, and it looks exactly like the structure that everyone gets at 0 millivolts. So that's kind of interesting, but it's also very comforting. And then here's a right side out structure. And like you heard from Shiva, you get a superposition of a mixture of different structures. Our superposition looks pretty ridiculously complicated. It does not look like the standard the transmembrane region, no doubt because we have a mixture of various structures. And so if we look at slices through the maps, here's the inside out channel, and here you see the S4, S5 linkers at this particular slice through the map. And there are the S4-5 linkers, and these are the bundle of S6 helices, and it's making an open pore. And here is just confusing. We have the superposition of many different structures, and the hope is that we can, using these maximum likelihood programs that now people who know how to write programs well have made really good versions of them, we may be able to tease out the various 3D structures that are underlying this. But coming back to Shiva's work in the past, that is 2 years ago, he did a structure of the EAG channel, again, non-conducting, but watch the voltage sensor do 2 clicks. That is, if we look at the depolarized structure, here is K1, R2, R3, those 3 basic residues, and then if you make the membrane potential negative, R2 and R3 have moved down below the charge transfer center. And you can ask, well, okay, so we're watching alpha helix move about 2 turns, about 10 angstroms. How is this going to be equivalent to moving these charges all the way across the membrane? Well, one thing to keep in mind is that above, at the top and at the bottom of the voltage sensor are water-filled regions. And so I've shown here kind of a rough estimate of the boundaries of water here on the extracellular side, here on the intracellular side. And the water then, having a high dielectric constant, basically conducts the membrane potential down into this cavity. The other thing is, notice here's K1, here's R2, R3, the side chains are all pointing up. And then when these pass by the charge transfer center, they're all pointing down. And this isn't the electric field pulling them around, it's the presence of water making these side chains orient either up or down. So we're getting not only the displacement of the S4 helix, but we're also getting a flip of the side chains as they move from above to below the charge transfer center. So that also gives us more charge than you would expect. There's one other thing going on, which is that, remember there was this pattern of a basic residue every 3rd residue in S4. Well, part of the S4 helix is actually a so-called 310 helix, which has just 3 residues per turn. And so that aligns the side chains on one side of the helix. And so it is really cute then that the S4 helix can switch between regular alpha helix 3.6 residues per turn to 3 residues per turn to help match the geometry as the helix slides past the charge transfer center. Okay, that's all I wanted to say. It's time for beer. But this is just to say that it's been a great privilege for me to be in this field and watch things go from, are there really channels, to here's the structure of a channel and here's how voltage sensors seem to work. The little bit of data that I did show you from our lab, these were images taken and processed by Youngyu Wu and Yosan and Yang did the biochemistry reconstitution, and Hideki really helped us develop this cryo-EM specimen. Thank you.